This is the second part of lecture 1. When I ended the previous part, I talked to you about top-down and bottom-up approaches. So experimentally, top-down approaches involve surveying systems to measure many changes simultaneously. This type of surveying type experiments has now gotten a label called omics. This is an approach most often experimental that measures many individual entities that make up a system simultaneously. The term omics is currently an overused and sometimes abused terms, but nevertheless it's useful to sort of define systems biology type experiments. So of course I talked to you at length about genomics. Genomics is the study of most and sometimes all genes involved in a certain physiological function and sometimes even in the functioning of a whole organism. The experimental genomics uses DNA sequencing and these microarray chips to study many changes simultaneously. Proteomics, in contrast, studies many proteins at the same time. It's harder to have all these protein changes clearly associated with a specific function, but nevertheless newer studies are starting to do this quite successfully, and proteomics just measures changes in the levels of proteins, and sometimes it also measures changes in the levels of the state of the protein, such as changes in state of phosphorylation or changes in state of acetylation or whatever. Most often proteomics uses mass spectrometry to identify the proteins, but there are also protein arrays using antibodies to known proteins and so on. Metabolomics is the study of all the metabolites in an organism, tissue or cells, and most often these metabolites, be it say lipids or sugars or nucleotides, are studied as in the context of a cell or tissue type and a perturbation, and most often metabolomics involves the use of liquid chromatography and mass spectrometry. So these large survey type experiments provide a lot of information, a lot of data, and these data need to be organized in a way that can be that will allow us to extract knowledge from the data sets, and the field that sort of deals with organizing all these large data sets is called bioinformatics. Basically, there can be databases of human genes, mouse genes, gene expression patterns. Many of these databases are freely available on PubMed. They are often searchable, and often the data in these data sets can be used for computational analysis. And this sort of, the usage for computational analysis is sort of a key feature of bioinformatic databases. Some commonly used databases include mRNA profiling, all the microarray data sets in GEO, the Gene Expression Omnibus, all characteristics of currently known proteins such as in SwissProt, or disease genes in OMIM, which allows you to identify mutations in genes that are associated with various diseases, or PharmGKB, that gives you all sorts of information about drugs for various therapeutic purposes. This is just, again, a very small subset of the databases that are available. You can peruse through the web and find many more databases as you need. A key characteristic of all of these databases is the ability to compute with these databases. What that means is that one can analyze this database and extract information with regarding to the data in the database. So what kind of computations can one do? One can do statistical correlation between entities in a database. For example, look at all the mRNAs that are expressed under a specified condition or specified perturbation or a specified time point. One can do statistical correlation between databases. So for example, you can combine the OMIM and GEO database and look at the effect of mutated genes in OMIM and the mRNA expression patterns in diseases and try to figure out what may be the relationship with mutation of specific genes and large-scale gene expression patterns. From these types of sort of statistical correlation analysis, and this is sort of a very important starting point, one can generate lists of genes, proteins, microRNAs that can be related to a specific physiological or pathophysiological state, or in the case of understanding regulation, how a perturbation changes a certain expression profile. And these data sets, these sort of lists form the basis for understanding parts of the system of a whole that might be involved in a specific function. From these lists, what can we do? We can build networks. What does the term network mean? Networks are really representations of systems such as genes or proteins and relationships such as interactions between the genes. These may be direct interactions between proteins. So protein-protein interactions are indeed called protein-protein interaction networks, or genes that regulate one another are called gene regulatory network. The entities are called nodes and the relationships are called edges. These are just terms that are used in the mathematical field that involves the study of networks. Networks are computable systems, and the computation can provide knowledge of how the system is organized. The branch of mathematics that deals with the computation of network is called graph theory, and people may refer to this, and I may refer to it also as the course goes by. So graph theory and network analysis are almost synonymous and can be used interchangeably. In contrast to network models that deal with top-down data, one can also build models with bottom-up modeling, and here the approach is to understand how systems are put together by identifying one component at a time and studying the binary relationship between these components. Many signaling pathways actually, which you can think of as small systems, have been identified this way. Dynamical models, and I will explain these terms as we go along, which are based on differential equations and can quantitatively estimate how these pathways respond to stimuli. And this kind of models that compute the response of pathways with respect to time, or sometimes even time and space, tell us how information is received, processed, and propagated within biological systems. So for instance, one can figure out for a certain level of signal, which really means concentration of hormone, what fraction of the receptors are occupied and how much does this receptor occupancy recept an intracellular signal to change the behavior of the cell, say by changing the activity of a channel or metabolic enzymes and so on. So let me summarize sort of the key ideas I've suggested in what systems biology is and these preliminary definitions that we will use throughout the course. Systems biology builds on physiology, molecular biology, biochemistry, and cell biology to understand how molecular components interact with one another to form functional units. Experiments in systems biology often involve measuring many cellular entities such as genes, proteins, lipids simultaneously, and these type of experiments are called omic experiments. Systems biology integrates experiments. A key feature of systems biology is the integration of experiments and computational modeling to understand how the system functions. This really is sort of the central theme or the central focus of systems biology. Within this, there are many, many variations, but understanding how systems function in terms of components and interaction is what systems biology really is. The use of computation, of course, is a key feature of systems biology, and here systems biology, even mammalian cellular systems biology, as compared to classical biological disciplines such as molecular biology, biochemistry, and cell biology, use is far more reliant on computational methodologies, and hence understanding how biological systems can be represented in computational terms and how we sort of compute or calculate the behavior of systems is going to be sort of an important driving theme that I will sort of keep coming back to during the course. Thank you.